your boy, Jericho Martinez again, and we're gonna learn about something cool today. We're gonna learn about amino acids. Woo! Let me tell you what an amino acid is. It contains a couple of groups, but this is one of the groups right here. An amino group and a carboxyl group. Both are linked to this central carbon atom called alpha carbon. Up here is also hydrogen, and right here is called the R group. As you know, amino acids have different properties. They could either be hydrophilic or hydrophobic. All right? So how would you know if it's either hydrophilic or hydrophobic? The difference is they're all the same. They all got that same amino group, the same carboxyl group, the same central carbon. But what differs between all of them and its properties are this R group. This is an R group, that's an R group, and that's an R group. You feel me? CH2, CH, CH3, CH3, that whole thing right there, that's an R group. And that tells whether this will be either hydrophobic or hydrophilic. As you can see, it differs here too. The R groups are different. Bring out this water. Oops. Bring out this water. It loves this water. This amino acid right here, it wants this water. It's hydrophilic. It wants it to rain on it. You feel me? This right here, you want to put water in it, it won't take it. It won't take this water. It says, don't touch me. So, the main lesson we should know here is all amino acids have something, and that's this right here, the amino group, this right here, carboxyl group, the central carbon atom, the hydrogen up here, and what differs and makes each different amino acids, which are 20 in proteins, is this right here, the R group. It differs for each amino acid. We're back now with our best friends, amino acid and amino acid. And you know what we're gonna do? We're gonna talk about polypeptide bonds. Alright, so we got amino acid right here, amino acid right here. They really love each other, so we're gonna make them go together. So the carboxyl group of one amino acid and the amino group of the other amino acid will join together, bang, bang, in a dehydration reaction. That means we're sucking the water out of it. We're dehydrating it. We're gonna make it thirsty, you feel me? And that results in what we like to call a peptide bond. Now, if we want to keep adding more of these amino chains to it, we do the same process. The carboxyl group is added to the amino group of the other amino acid. So amino acid plus amino acid. So we could keep making links to these amino acids, and that results in something called a polypeptide bond. More than two amino acids together can form a polypeptide bond. Okay, class, one more thing. Now, to release the amino acids of a polypeptide bond is to do the opposite. Not through a dehydration reaction, it's called hydrolysis. And by adding water to a polypeptide bond, you can release the amino acid.
Alright, so what's going on? I'm on the beach right now, but I figured, hey, why not talk about a protein shape and how it determines its function? I mean, why not? Alright, so we have 8 functions of a protein. I'm going to talk about all of them.
We have enzymes first. They speed up and regulate reactions. Then we have structural proteins. They connect tissues to make things like hair and tendons. And we have contractile proteins that make up muscles. Defensive, things like antibodies. They protect our bodies from disease, illnesses, whatever. And we have signal proteins, things like they're hormones that pretty much communicate bodily functions, tell our body what to do. Then we have receptor proteins that transmit bodily signals into cells. And we have transport proteins that act as movers, things like hemoglobin deliver oxygen throughout the body. Then we have storage proteins, things like egg albumin. So those are the functions. Pretty much a shape determines the specific function, and there's two types of shapes. There's a general shape, which is things like globular and fibrous, and then there's also a specific shape, which refers to the specific like coils and folds, grooves in a protein itself. And there's also a process called denaturation, and pretty much what that is it's a change in heat, pH, or salt concentration. And when that happens, it changes the structure of the protein, which therefore changes the function of the protein. So that's pretty much it. That is all about protein structure.
There are structures to a protein. The primary, secondary, tertiary, and quaternary. In the primary structure, it is a linear sequence of amino acids. There are various amino acids, for example. Secondary structure is an alpha helix and beta pleated sheet formed by hydrogen bonds between atoms of the polypeptide backbone, as shown here. And this is an amino acid in the beta pleated sheet. Tertiary structure is a buildup from the primary and secondary and is a three-dimensional shape built up from the R groups. The tertiary structure is a polypeptide, and it forms a quaternary structure when two polypeptides form together. This is one and this is two together, so it's the quaternary structure of a protein.